Okay.
So in this project, in this challenge, your goal is to overexpress a particular gene to overproduce glucosinolates,
Yes.
a type of secondary metabolites.
Secondary.
Plant secondary.
Yes.
So what are plant secondary metabolites?
Well, what I understood is that it's not the, since they are not primary metabolites, it has to do with the pathway and the biosynthesis of the glucosinolates, no? And also I saw that the glucosinolates were for plant defense, so
So
maybe
you can
those
say the secondary
they are
metabolites are mainly for plant defense.
Yes.
That's why it's secondary. Only they need to defend themselves, only then they produce
Yes.
that particular metabolite. And what type of benefits we get from those glucosinolates, human benefits?
I saw that they were chemoprotective, so
What does it mean?
it means that they can be used to stop or for the expression of cancer. So I saw that they use it in pharma, the glucosinolates.
So they are anti-carcinogenic
Yes.
and anti-inflammatory, because inflammation is the main cause of cancer.
Of cancer.
Okay, and which gene you chose for the expression?
Mine was MYB28, and it produced the overexpression in the aliphatic pathway.
Okay. And which vector?
I use PBI 121, I believe.
Okay.
It had CAM V25S,
What is that?
the promoter.
The promoter. Okay.
It has the reporter gene GOS. Also, it used kenamycin,
What is that?
the marker gene.
Okay. So what is the difference between marker gene and reporter gene?
Well, the marker gene is it's the one that we use to see if there's to use it in the plate, so we can know if it has or has not the expression of, well, when you insert it and then you make it growth and then you use that one to see if it grows or not.
So that means it's a transformed cells.
Yes.
Okay. Transformed cells you can select. That's why it's called selectable marker.
Selectable marker,
The
yes.
non-transformed cells will die. Okay. And the reporter gene?
Gene. I can't remember.
The reporter gene is to know whether the promoter is working well or not.
Yes.
Sometimes we use GFP, green fluorescent protein. So if the promoter is working well, that GFP will express and will fluoresce,
Yes.
because it will produce that GFP protein. Okay? So you can say that the promoter is working well. So theoretically, if the promoter is working well, so promoter, GFP, and after that your gene. So if the promoter will be working well, it will express the GFP as well as your gene.
Okay.
Okay, and the final question, what is plant transformation?
Well, in my case, I use Agrobacterium tumefaciens, and what I did was after the transformation.
Transformation?
Yes, of the vector, and it's already in the E. coli cells. Then you have to insert it into the Agrobacterium, so that since it's like the growth of the plant, you have to insert it, and then with the tDNA.
So who will insert? The Agrobacterium.
The Agrobacterium.
They have specific characteristics. They insert your gene of interest
into the plant genome.
Okay, so that it will start to express.
Yes.
Okay, thank you very much. That's all.